Good morning and good afternoon, everyone. This is Catherine Gravlin from the IQ Secretariat. I'd like to thank you for joining us this morning's webinar entitled Focus on Downregulation, CYP2C Induction, and CYP2B6 Positive Control. This webinar is brought to you by the IQ Consortium Induction Working Group. Before I turn things over to Donald Tweedy, who will introduce our presenter, I'd like to take care of a few housekeeping items. First, you'll see a panel on the right-hand side of your screen. Clicking on this arrow, you will collapse or expand the control panel. Next, taking the following steps will help greatly reduce audio feedback. One, if you're a VIP participant, please use a headset and keep wireless devices away from the computer. Participants calling in by the telephone, please enter the audio PIN number shown here. I'm sorry, apologies. Next, to submit questions, enter in the questions module in the control panel, highlighted in red here during and after the presentations. And finally, as a reminder, this webinar is subject to antitrust guidelines adopted by the IQ Consortium Board of Directors. These guidelines require us to stay away from discussion of any commercially sensitive topics such as pricing and market shares. We will be recording this webinar and we will be posting it 3 hours after we complete this. And then we will send out the part 2 invitation as well. At this time, I will turn it over to Donald, who will now introduce our presenter.
Great, thank you. So our presenter this morning is Dr. Naresh Hariprasad, who's a director at Vertex Pharmaceuticals. He received his B.Pharm and Masters in Medical Science from the University of KwaZulu-Natal in South Africa and received his PhD from the University of Cincinnati. And he joined Vertex in 2010. He has functioned as the chair of the Induction Working Group since 2014 and is a lead author on the first Induction Working Group white paper, which will be the focus of today's presentation. I also wanted to make a couple of other introductions. So available as a panelist for answering questions at the end of today's webinar is Diane Ramsden, who's a senior scientist in early development at Alnylam Pharmaceuticals. She received a BS from University of Connecticut, and she's currently responsible for representing drug metabolism and pharmacokinetics and program teams, and for designing mechanistic ADME studies. Prior to joining Alnylam, Diane worked with me for 16 years at Boehringer Ingelheim, where she served as a DMPK project rep, and was the head of our hepatocyte lab and our go-to person for induction studies, and has been a core member of the Induction Working Group since it was established. She's also the lead author in a commentary which is being composed just now, and we hope to get out by the beginning of next year, focusing on some of the variability we see in clinical studies on induction. Also Jane Kenny, who wasn't able to be with us today. Just a brief introduction on Jane, who's been a key contributor. She's an associate director and principal scientist in DMPK at Genentech, received a PhD from the University of Liverpool, and has over 15 years of drug discovery and development experience. So she led the data interpretation subgroup, and that will be the focus of the second, which was the focus of the second white paper published in DMD, and will be the topic for the second webinar that Naresh will introduce at the end of his presentation. I have been interested in this topic for a number of years, and have been involved with the Induction Working Group since its inception. And my interest is really in ensuring that we have effective interactions with regulators to drive towards a scientifically defensible position on drug-drug interactions. And from my perspective, this entails an understanding of regulators' thinking and what they're looking for, as well as pushing for a pragmatic industry perspective so that we can minimize the need for studies that don't add value, particularly clinical studies, but also ensure that we maintain patient safety. Now, the genesis of this induction working group really came out of a Q&A session at the International ISIS-X meeting in Toronto in 2013, where industry were providing some very specific feedback to the presenter from the EMA. And then when questioned, the presenter, Eva Berglund, was very receptive to a proposal from industry that we could provide more of a data-driven approach to support some specific alternative recommendations on the EMA DDI guidance, which at times was fairly prescriptive. We then approached the IQ Drug Metabolism Leadership Group, who are now the Translational and ADME Sciences Leadership Group, to form a committee that could interact directly with regulators and also generate white papers. We came up with a short list of specific feedback, and a lot of that feedback will be addressed in the next couple of webinars. There are additional 2 publications planned for the induction working group. One is a time course for in vitro induction and data analysis, and the other I mentioned, which is the commentary on the variability in clinical induction studies. We very much encourage feedback, both positive and negative, and so please, whether through today's Q&A or through feedback by email, your thoughts on today's presentation or on any of the induction working group publications is encouraged and very welcome. And now I'll pass over to Naresh for the presentation.
Donald, thank you for that introduction, and thank you for highlighting how the group was formed. And I'm going to touch a little more on that as well, give a little more details related to that in terms of the genesis of the group / makeup of the group and all of the work that we've done up to this point. So as was pointed out by Donald and Catherine as well, the focus of today's webinar will be on our first publication related to downregulation in vitro, as well as assessment of CYP2C induction. And when I talk about CYP2C induction, I'm going to be focusing on CYP2C8, CYP2C9, CYP2C19, and some of the challenges associated with conducting in vitro studies for CYP2C induction. And finally, I'm going to close off by talking about the recommendation to use CYTCO as a positive control and some of the data that we generated to support a recommendation to use another positive control or potentially use another positive control, which is phenobarbital. So I'm going to take, excuse me. So as was pointed out by Donald, one of the key challenges or one of the key objectives for us was to move towards a better understanding of the overall regulatory requirements for drug-drug interactions, but primarily focused on induction. And as I talk through the slides today, and you'll hear in our second webinar as well, we didn't just focus on CYP3A4. We also looked at other sub-pathways as well. And as was pointed out by Donald, we want to make sure that we take a data-driven approach to working with regulatory agencies to update their requirements. And very often this meant that, when we couldn't find data in the literature, we actually took that additional step and generated data within the induction working group to sort of emphasize or strengthen our recommendations. So on this slide, I wanted to highlight the fact that we have a diverse group of members, members from large pharmaceutical companies as well as mid-sized pharmaceutical companies. You can say there's about 21 names here. In addition to having individuals from pharmaceutical companies, we also thought it was important that we include members from CROs as well, because a lot of companies nowadays outsource their induction studies. And you'll note that we have a representative from 2 CROs as members of this team, one from Corning, the other from Xenotech. The one thing I also wanted to highlight is the fact that, when we started out this process as was pointed out by Donald, we had about 13 different questions that we wanted to use as a discussion point with regulatory agencies. And initially, that entailed discussions primarily with the EMA, but that has evolved now to include discussions with the FDA as well. As we went through this process, what we noted was that we needed to break this larger sort of group of team members into smaller groups. So you'll note that, for example, we have sub-teams that were led by individuals. For example, Jane Kenny led the data interpretation sub-team, and Jane and Diane will be presenting the second webinar in this series. We also have a sub-team that is looking into time cost for induction, et cetera, et cetera. But the key thing to note was that even though we had these sub-teams, we had several levels of reviews. So even though the sub-team came up with the recommendations, we also reviewed recommendations / data, et cetera, from the sub-teams as a larger working group. And in addition to that, we also had recommendations reviewed by the drug metabolism leadership group as well, just to make sure there was consensus in terms of the recommendations that we came up with. So on this slide, you'll note that we have a series of manuscripts, two that have already been published and are in the literature. So the first white paper, which I'm going to talk about today, was published in DMD, and more recently, our second white paper was published. And as was pointed out by Donald, we are working on a clinical commentary related to variability that was observed, and this came out from our analysis of the data that was put together for the second white paper. And finally, there is a third white paper that we're working on, because there are a lot of questions and challenges related to data fitting. And in addition to focusing on data fitting, we also going to present work related to time cost for induction. So, for example, the EMA requests that in vitro induction studies be conducted over 72 hours. We actually conducted multiple studies to assess whether shorter incubation times would be sufficient to provide usable data to assess induction risk. So the last time that a survey was conducted to look at how in vitro induction studies were conducted was in 2003, and this was an initiative that was conducted by pharma, and I believe the first author was Bjornsson. And essentially, since that 2003 publication, the DDI guidelines have been updated. So we felt that it was timely for us to do a review or a survey of how induction studies are currently being conducted. So the manner in which we collected information to assess how individual companies are conducting induction studies is we sent out a survey or a questionnaire using Excel. In that, we provided a set of questions that were either open, close-ended, or multiple choice. And in addition to collecting in vitro data, we also collected clinical data as well, because it was important for us to assess in vitro to in vivo correlation. More importantly, in addition to collecting induction data for proprietary compounds, we also encourage companies to provide us with data for non-proprietary compounds as well. And again, when we did the survey, it was blinded, and you'll note that in subsequent slides, I represent data from individual companies as company ABC or hepatocyte lot ABC, so that data can't be tracked back to individual companies. The other important thing we did, and an important finding for us, was we collected data for individual lots of hepatocytes because we also wanted to see the extent of variability of the induction response across different companies. And finally, we also, using SurveyMonkey, asked individuals about the types of questions they are getting from regulators. And so all of that information helped us to pull together the first manuscript, which focused on the assessment of downregulation, in vitro assessment of CYP2C induction, and use of CYP2 as a CYP2B6 positive control. So as I pointed out, we asked individual companies to give us a sense of the types of questions they're getting from regulators. And interestingly enough, of all the companies that responded to the survey, 4 companies indicated that they got questions related to downregulation. So in this slide, I'm going to start talking about some of the responses that we received related to downregulation. So the first thing we wanted to look into was how individual companies are conducting the in vitro induction studies, because that could potentially impact the downregulation that is observed in vitro. What we noted was that for the most part, companies either used the monoculture system. In fact, most companies use the monoculture system, but there is a fair proportion of companies that are also using the sandwich culture for the assessment of induction. In terms of well format, we noted that for the most part, companies have evolved and are using 96 well plates to assess induction, and that essentially gives them a higher throughput. And just from personal experience, what I've noted is that assessment of induction in 96 well format is as good as assessment of induction in 24, 48 well format. There were some initial findings or some concerns related to platability in 96 well versus 24 well versus 6 well. But from all the data I've seen, the data correlate very well between these different formats. There are certainly some companies who are still using 24 well format, and a fair proportion of individual companies still using the 48 well plate format. In terms of incubation time, we thought this was important to information to collect as well, because we know that the longer the cells are in culture, there is potential that cells could be stressed, and that could be a factor that could contribute to downregulation that is observed in vitro. Interestingly enough, and this is not surprising, of the 17 companies that responded to this question, most companies did their induction assessment over 48-hour time period, while some companies still conducted their assessment using the incubation 72-hour incubation time. And finally, just again related to this evaluation of downregulation was we also asked companies whether they assessed cytotoxicity and how they assessed cytotoxicity. And the response was really interesting. For the most part, the assessment of cytotoxicity is done using or looking at the morphology of cells. And I think that's somewhat subjective, but that was an interesting response. Other companies used viability, and others used LDH assessment of the tool as well, kit as well. So, this was a starting point for us to get a good sort of overview of how individual companies are conducting their induction studies and how some of this could impact the downregulation that may be observed in vitro. So on the next slide, I'm going to go into some of the specifics associated with the observations of downregulation. And what's interesting as a starting point is to note that, 10 to 20% of respondents observed downregulation during the in vitro induction studies when they screened their compounds for induction risk. So this was really interesting, and it's not too surprising actually. There was a recent publication by Badolo et al., where they looked at about 309 compounds, and I believe about 10 to 20% of their compounds, of the 300 odd compounds, there was this observation of downregulation during their assessment as well. Interestingly enough, for the most part, individuals, when they assess induction, they generally assess induction using looking at CYP1A2, CYP2B6, CYP3A4. But some companies, in addition to assessing induction of those CYP enzymes, also looked at induction risk related to CYP2C and UGT1A1 as well. And you'll note that these are the enzymes that were impacted when it came to downregulation as well, right? And again, also of interest was the fact that of the companies that observed downregulation, many saw downregulation of about, ranging from about 0.1 to 0.3 fold. And this is fold relative to the DMSO controller, the vehicle that is used. So that was another interesting observation as well. And added to that was the fact that there was a fair number of companies who saw downregulation of less than 0.1 fold. And many of the respondents who saw this reduction in induction fold also noted that it was concentration dependent as well. So this pie chart highlights the mRNA reduction levels, but in parallel to that, many companies also looked at enzyme activity as well. And interestingly enough, even though enzyme activity is not as sensitive as mRNA, many companies, in addition to observing a reduction in mRNA and observing a concentration-dependent reduction in mRNA levels, also saw a reduction in enzyme levels as well. So this gives you a sense of the fact that downregulation is being observed. What was another important point to note is the fact that companies did do an evaluation of whether there's a correlation related to a structural series, therapeutic area, or compound class, and all respondents indicated that there was no correlation. So it wasn't like a specific subset of compounds were known to cause or would always cause downregulation as opposed to another subset of compounds. Before I get into the recommendation, I also wanted to touch on a couple of other issues. I did touch on cytotoxicity as a reason for potentially observing downregulation. But the other factor to consider is inhibition as well, specifically time-dependent inhibition. What was interesting from the survey results as well was that most companies didn't have any established criteria, when they observed downregulation in vitro. So, for example, only 9 of the 17 companies had cutoff criteria when they for downregulation observed in vitro and then potential follow-up, but most companies did not have that. Of the survey respondents, 3 companies who did observe downregulation in vitro did follow-up in vivo and mechanistic studies to understand the reason for downregulation. In fact, of those 3 companies, 2 did follow up and do clinical studies to assess the risk for downregulation. And interestingly enough, of those, even though the downregulation was observed in vitro, they did not observe the same extent of downregulation or any inhibition in the clinic itself. So I'm going to jump into the recommendations from the IQ here, the IQ working group. one of the key things to note here is that, even though we have, and different companies are observing downregulation in vitro, there's no positive control compound. So essentially, when we do induction studies, we have a positive control for CYP3A4, for CYP2B6, for CYP1A2, and we can then understand how well the system is working. Is it an optimal system to assess induction? But on the flip side, we don't really have a good positive control to assess whether the monoculture system or the sandwich culture system is a good system or in vitro tool to assess downregulation. So we did follow up and have a discussion with the EMA regarding this, regarding the fact that it would be useful for us to have a good positive control to assess the quality of the in vitro system or validate the in vitro system. They did point out to us that they are aware of one example, but there's no, during our discussion, did not give us any sort of information regarding that compound. And to my knowledge, there's no publication in the literature which talks about a small molecule compound where downregulation is observed in the clinic. So, there's that challenge. And so, a couple of things to consider then is, any sort of downregulation that is observed in vitro should be considered with caution. Obviously, when downregulation is observed, it's important to understand cytotoxicity, which I've highlighted a couple of times. It's important to understand whether the compound that you're assessing is a time-dependent inhibitor. In fact, when I presented some of our findings at the ISSX, one potential recommendation that came up was, in terms of cytotoxicity, it may be good to use more than one kit to assess cytotoxicity so that you potentially pick up, for example, apoptosis and necrosis, et cetera, et cetera. So again, it's hard to make a hard and fast recommendation regarding downregulation just because of the fact that there's no good positive control that can be used in vitro and where an observation of inhibition was observed in the clinic. I guess the one example which has been touted is colchicine. And even though colchicine does cause significant downregulation in vitro, inhibition has not been observed in the clinic, and that may be because of the exposure levels. So for example, in the in vitro system, you're using high concentration of compound, whereas when you dose the compound in the clinic, you may not be able to attain the levels that you would in an in vitro system. So now I'm going to move on and talk about CYP2C induction. So in terms of CYP2C induction, there are some real challenges associated with assessment of 2C induction in vitro. So what we did was as was pointed out earlier in the webinar, we collected data from several companies, and represented on this slide the data that we obtained from 8 different respondents. And you'll note that what's being presented here is data for CYP2C9 mRNA. And right next to the CYP2C9 data, we also present data for CYP3A4 as well. Now, I've been involved in conducting and helping to interpret induction data for several years now. And over the years, what I've noted is that, generally, when it comes to CYP2C induction, the fold that you observe in vitro is low. And for the most part, from the data that I've seen, even with a compound that has a very potent and selective PXR transactivator, like rifampicin, generally the fold response is 4-fold or lower. So that makes interpretation of the data very challenging. If your positive control is giving you a 4-fold inductive response. The other point to note as well is that, this 4-fold response with CYP2C induction is highly variable across the different lots of hepatocytes that are used and the different companies as well. Interestingly enough, and not surprisingly, even though you get a fourfold or less than fourfold inductive response in CYP2C9, generally you'd get a robust inductive response with CYP3A4, and this is fairly consistent in the literature as well. So the one thing we wanted to make sure of was that, you know, we want to make sure we were not seeing this variability or lack of inductive response because of protocol differences between different companies. So what we then, as a follow-up, did was we collected data from one company, but data was then generated in 10 different lots of cryopreserved human hepatocytes. And on this slide, you can see data for CYP3A4, CYP2C8, CYP2C9, and CYP2C19 as well. And again, you're getting this nice, robust inductive response in all 10 lots of hepatocytes that we used to assess CYP3A4 induction. You're getting a nice sort of fold change in mRNA as well as in activity as well. But you know, the one thing to note here is you know, the axes are different. So for example, for CYP3A4, we have our axis, our y-axis going all the way up to 80-fold, whereas for the for CYP2C8, CYP2C9, CYP2C19, our axis goes up to 20-fold. And again, you can see this consistent sort of message that you know, the fold induction is not as robust as is observed for CYP3A4. What is interesting to note, though, is that for CYP2C8 and CYP2C9, you're getting a nice fold increase in mRNA and a consistently nice sort of correlative increase in activity as well. So this is fairly consistent for CYP2C8 and CYP2C9, even though the fold changes are lower. Not surprisingly, for CYP2C19, the activity change is higher than the mRNA change. And this may be related to the substrate that's being used to assess functional activity for CYP2C19. So the one thing, you know, when as we went through these data, the one thing we noted was this nice correlation that we're seeing between fold changes in mRNA of CYP3A4 and CYP2C9, CYP2C8, etc. And you know, if you look at our paper, we actually do these correlation analyses where we plot out the fold inductions for CYP3A4, CYP2C9, or CYP2C8, and we see again this nice correlation. And please take a look at our manuscript. I didn't include that data in this presentation. But what I wanted to highlight was, you know, and drive towards was a recommendation on how to interpret 2C data, even though you have a limited induction window based on in vitro data. So I'm switching a little bit on this slide and looking at this table to some clinical observations, drug-drug interactions with CYP2C perpetrator compounds. And the one that is most significant would be the rifampicin interaction with warfarin, right? And warfarin, for the most part, everybody knows it's an anticoagulant, so you want to make sure that you have the right sort of doses because it impacts prothrombin time, which could obviously impact the anticoagulant effect, which necessitates therapeutic drug monitoring. So a lot of times, you know, if you co-administer rifampicin with a compound like warfarin, there's a high risk that you would get a fairly significant drug-drug interaction, which could be life-threatening. The one thing to also note is the fact that even though there are DDIs with multiple perpetrator compounds, aprepitant, ritonavir, bosantin, and you can keep going down the list. Most of the interactions that you would see tend to be on the weak to moderate side, okay? So on this slide, I just wanted to re-emphasize the fact that for many of the compounds that are CYP2C inducers, there are data available in the clinic which indicate, which give you a sense of the inductive response for CYP3A as well. And you'll see again, right, with rifampicin, for example, with the probe substrate like simvastatin, you're getting a 91% decrease. With bosantin, similarly, you're getting a 69% decrease. So essentially, the message that we're trying to get to is that, you know, given the co-regulatory pathways for CYP2C and CYP3A4, so we know that generally pregnane X receptor, constitutive androsten receptor are involved in the regulation of CYP2C as well as CYP3A4. The message we're trying to drive to is that just given this correlation, this positive correlation in terms of the inductive response between CYP3A and the different CYP2Cs, it may be possible, or it is possible, to make a recommendation on DDI risk based on CYP3A4 data, induction data. So again, on this slide, we re-emphasize the fact that with perpetrators like aprepitant, bosantin, carbamazepine, rifampicin, you can see induction that's observed, for example, with aprepitant, where you get a reduction of about 31% reduction in AUC when you co-administer aprepitant with a compound that's a CYP2C substrate. You get a reduction in AUC of about 22% when you administer a compound that's a substrate of CYP3A4. And if you keep going down the list, you'll see that for the most part, when you see induction for CYP2C related to CYP2C, you see a consistent reduction for CYP3A as well. So in general, as I pointed out in the initial table, the extent of induction for CYP3A tends to be greater than for CYP2C. There are exceptions to this, and this may be related to mixed mechanism perpetrators like ritonavir. So here, even though you get a reduction in AUC for CYP2C, you're seeing inhibition of CYP3A, so that one will be tough to assess. In general, there's this consistency in terms of induction response for CYP3A and CYP2C. So I'm going to talk about the recommendations now from the IQ induction working group. So I've pointed it out several times now, the fact that, we are getting a low and variable response for in vitro induction using human hepatocytes. Ideally, as the field evolves, when new in vitro tools become available, like co-cultured systems, etc., it may be possible to get a larger window with positive control compounds. But up until that point, our recommendation is that induction of CYP3A4 can be used as a sensitive marker of the potential for induction of CYP2C isoforms. So a compound that's determined to have a low risk to cause a 3A4 induction-based DDI will very likely have a low risk to cause a CYP2C induction-based drug-drug interaction. I'm going to speed things up a little bit here just to make sure we have enough time for Q&A, and talk quickly about the data that we pulled together related to CYTCO and phenobarbital. So the question or the recommendation from the EMA is to use CYTCO as your positive control to assess 2B6 induction. So there are several companies who primarily use phenobarbital as a positive control compound. So what we did was we pulled together data from several different companies We present the fold induction response in the same lots of hepatocytes. And these are data for 26 different lots of hepatocytes. And clearly, the take-home message from this sort of graph or slide indicates that the inductive response observed with phenobarbital is higher than the inductive in general, higher than the inductive response that is observed with CYP2. You'll note that in some lots of hepatocytes, the CYP2 inductive response is closer to twofold, whereas in that same lot of hepatocytes, the inductive response due to phenobarbital is about 5 to 7-fold. And again, everybody in the working group was aware of the fact that in addition to phenobarbital being a CAR activator, it is known that phenobarbital is also a PXR transactivator, while CYP2 at the concentration that is indicated on the slide is not a PXR transactivator. So in terms of recommendations, the working group recommends that instead of just using CYP2 as the CYP2B6 positive control, we'd recommend that regulatory agencies accept either CYP2 or phenobarbital as a positive control for CYP2B6 induction. So the other thing I wanted to point out was the fact that, given the fact that induction is assessed using 3 different lots of hepatocytes, the IQ induction working group thought that missing induction because of CAR transactivation across all 3 lots of hepatocytes would be really low. I'm going to jump to the next slide and highlight the immense amount of work that's been done to understand or to work on data interpretation of in vitro induction data. And we've actually pulled together a fair amount of in vitro data and clinical induction data sets, and that includes about 51 compounds. And I think, from what I've seen in the literature, I don't think anybody has pulled together as much in vitro and clinical data. As I pointed out earlier, in addition to collating and pulling together data for proprietary compounds, we also pull together data for non-proprietary compounds as well. And as I highlighted in my earlier slide where I talked about the different manuscripts that have been published, I also wanted to highlight the fact that we'll have a commentary coming out. We're hoping to publish a commentary early next year related to clinical variability. Many companies struggle or have challenges with fitting data. So we're hoping to pull together a manuscript which would provide some recommendations on how to fit data for compounds where an Emax is not observed or a bell-shaped response is observed. So I wanted to stop at this point just to make sure, and I did rush through some of the later slides. I just want to make sure we have enough time for Q&A. Thank you.
Thanks, Naresh. And just as a reminder to participants, you can go in online and pose questions that we can address now. So I'm going to try and manage the questions a little bit, maybe take them in order of your presentation, Naresh.
Sure.
So for downregulation, would cytotoxicity not be reflected in more of a broad spectrum effect? In other words, you'd see a decrease in multiple endpoints. And so when you showed the data for the various CYPs and UGT, were they all downregulated, or was it specific for a particular CYP?
So in certain instances, it was specific for certain SIPs, but there were survey respondents who indicated that in addition to observing downregulation for CYP3A4, for example, there was also downregulation of UGT1A1, CYP2B6, etc. There was also an indication that there was differential downregulation observed. So, for example, in certain instances, even though 3A4 was downregulated, 2B wasn't downregulated. And that may just be related to the regulatory pathways involved in induction. I don't know if Diane wanted to add to that.
Sorry, I was having trouble unmuting myself. Yeah, I think in some cases the respondents indicated that there was a global effect, and this could be linked to a really late cytotoxic effect, so the hepatocytes just were really struggling. But in my own experience, I have seen cases where cytotoxicity endpoints don't actually pick up the early cytotox events, and in some cases, at the transcript level or at GAPDH, it's picked up earlier. So I wouldn't necessarily say that consistently you would expect to see, in the case of cytotoxicity, all of the enzymes being downregulated. I think there is a temporal component to that.
Okay, so on that topic, there's another question on, does the induction working group have recommendations on how to measure cytotoxicity? And maybe I can add a personal perspective to this, which is certainly listening to and talking to regulators. This is a topic that comes up repeatedly, particularly from the EMA, who question whether or not we're being, as an industry, rigorous enough in our induction studies at assessing cytotoxicity.
Yeah, and I can add to that too. You know, one recommendation that came out of the discussions we had with individuals from different pharmaceutical companies, CROs, etc., was, you know, it may be worthwhile using more than one toolkit to assess induction. You know, for example, you know, as Diane was pointing out, you know, at certain concentrations you could get apoptosis. At higher concentrations you could get necrosis. So if you use a kit that only focuses on one, you'll be missing the other sort of cytotoxic events. So that may be something to consider. I mean, obviously there are challenges with execution then, right? Because some of the tools you can, you know, you have to plate a whole separate set of cells to assess cytotoxicity. So there are challenges, but there are some newer kits where, you know, there's no interference, where the same cells could be used to assess viability / cytotoxicity, and also the cells still remain useful to assess induction using mRNA or activity. Diane, please chime in if I missed anything.
No, I think the other thing to note is that this is really like a situational preference. A lot of companies have experience with specific assays and have a large database using those assays. So I think really it comes down to if there is a mechanistic response that needs to be probed a bit further, then additional assays could be included or more sensitive assays could be included. But for the most part, our standard kind of kits of cytotoxicity assessment during induction studies perform pretty well. I think it's just really, if we see this sort of observation, due diligence to kind of follow up with maybe some additional endpoint screens to see if this could be a cause prior to going into some additional mechanistic studies to try to identify what may be the reason for observing downregulation, for instance.
So as a follow-up to that, one of the questions is, you know, have there been observations of induction despite cytotoxicity? And in that case, how do you reliably measure induction? And I suppose my extension of that question is, are we underestimating induction in cases where we see some cytotoxicity?
Yeah, that's a really good question, and maybe challenging to answer. I'm going to do my best. I think I have seen examples where even though there is cytotoxicity, there is an inductive response. One approach could be to use a shorter incubation time. So, for example, instead of using a 72-hour incubation time, could you assess induction risk using a 48-hour incubation time, or an even shorter incubation time of 24 hours? But obviously, that would necessitate you having done some additional validation work with other sort of known inducers to make sure you can interpret your data. The other thing I've noted is that, some lots of hepatocytes tend to be more susceptible to cytotoxicity than other lots of hepatocytes. So, for example, if you conducted an assessment with 3 lots and you saw cytotoxicity in 1 lot, you could use an additional lot as well. So those are some of the approaches that you could use. I have seen some individuals recommending you adjust, for viability or cytotoxicity, and I'm not sure if that's good because, again, you need to develop some sort of an in vitro to in vivo correlation to do that. So from my viewpoint, I think, the 2 approaches I just discussed, maybe shorter incubation time or using additional lots of hepatocytes, may be useful. Please chime in, Diane or Donald, if there's other viewpoints.
Yeah, I think, can I just add one thing quickly about I think some of this discussion may be included in the data analysis and time course white paper, because often what you can see is kind of a bell-shaped curve, and that could be indicative of cytotoxicity at your higher test concentrations. So this is definitely something that is being discussed, and we're planning to propose some recommendations on how to deal with this type of data in the future.
Great. And in the interest of time, I've got a couple of questions on CYP2C, which I'm going to combine. So CYP2C induction is a moderate effect, given the variability seen by the induction working group and reported by Kenny et al. In the second white paper. Do we ever expect to be able to provide definitive induction data for CYP2Cs? And the second question, which is, can be addressed there as well, are there cases where evaluation of clinical CYP2C induction might be needed or recommended? And can any of the isoforms be evaluated, 2C8, 2C9, or 2C19? Is a tiered approach possible? Which would be the most sensitive?
So, there have been some attempts to improve the robustness of the induction response that is observed in vitro. I know there was one group that was trying to plate cells without dexamethasone because there's this belief that the glucocorticoid receptor is involved in induction, and so by adding dexamethasone to your incubation media, you could be sort of blunting the inductive response. I've tried that. The problem is the cells don't plate well, so the quality of data that you're getting may be questionable, but again, it is something that's being evaluated. There are newer tools coming out, co-cultured systems, microfluidic systems that may be better than the monoculture system. But I think there's just not enough data right now to make that recommendation or make any recommendations regarding which tool outside of the monocultured or sandwich culture system would be better. So right now it'd be challenging to assess this in vitro. Donald, does that cover?
Yeah, and then the question was, can any of the CYP isoforms be evaluated, and is there a tiered approach, or is one most sensitive? And when you take a look at your 2C19 data, it's quite remarkable that you had such a large increase in enzyme activity for CYP2C19 despite moderate increase in message, and that was a lot larger than any response seen from 2C8 and particularly for 2C9. So, what's the mechanism behind that, and would that give us false positives for induction of CYP2C8 and CYP2C9?
Yeah. I guess with CYP2C19, it may be related to the probe substrate that's being used. I'm drawing a blank. I think it's S-mefenatine that's being used. I could be wrong. But maybe, that substrate is also being cleared via another pathway like CYP3A4, and therefore the window looks larger. So my view, and again, any in vitro data will have to be correlated to clinical data. And even though there are some examples of CYP2C19 induction, I'm not sure if there's enough examples, clinical examples, enough clinical data to draw correlations for specific CYP2Cs. So again, it may be premature to do that right now.
Great. And we have 1 minute left, and I'm just wondering, maybe just from my perspective, I don't know whether people are aware that there was a recent IQ FDA roundtable where induction was one of the topics. Any take-home messages you want to share from that meeting with the FDA, Naresh?
Sure. It was a very constructive discussion. There were questions regarding the utilization of PBPK modeling. The take-home message was that, even though the FDA has these guidances / guidelines, they're open to having discussions about, how data are being interpreted. I think a lot of the work we've done and the knowledge that we have in industry, they may not be aware of, the kind of extensive sort of data that we may have collated or pulled together at an individual company to understand induction risk. So the take-home message was, they're open to discussion on, for example, when DDI risk is assessed, protein binding, the cutoff is 1%, but they're open to having a discussion about using a measured binding level. They are also looking at other systems to assess induction, like microphysiological system, co-cultured systems. So they're open to discussion and sharing of data regarding those systems as well. So those were two take-home messages for me.
Right. Well, we're out of time. I want to thank Naresh for the presentation and for Diane for joining in the questions, and for the participants. I hope you found this valuable. Catherine, I'll hand it back to you. Anything last minute before we close?
Nope, just want to thank everyone for participating and remind everyone that the part 2 will take place December 6th at 11 p.m. Eastern time. And we will post this webinar in approximately about 3 hours. So thank you again, everyone.
Great. Thank you.
Have a great day.
Thank you. Bye.